Hi all. So today I will tell you how you can actually memorize any topic very easily in the form of flowchart. So already we have done with all the topics in biology. Also I have taught you how to learn the lessons in an easy way. Today I will be showing you how easily you can learn any lesson through a simple flowchart like this, taking the first lesson as the example, that is nutrition lesson in biology. So let us go about and see how easily you can memorize. Okay, so when you are starting with such type of revision, you should be familiar with the topic already. So in the flowchart, whatever we are doing, we are trying to memorize whatever we have learned already in one paper. So like this, if you make these sort of flowcharts for every lesson and keep it ready during your exams, this is very useful actually to just browse through and go to the exam, so that it will be giving you a very quick revision of all the important points related to that lesson. So why to waste time? Let us go on and learn, okay, how to actually quickly revise your topics. So let us learn about the nutrition topic. So the nutrition topic, okay, you will be learning, okay, through this board. Only this board information is sufficient to learn your entire topic. So let us quickly go through what is written here. So nutrition, you know what is nutrition? Intake and utilization of the food by the organism. So that you will write down like that as a point. Okay, so that if a definition comes in the exam, you can write. Okay, so nutrition, you know, there are two types of nutrition depending upon how the organism will make its food. So autotroph or heterotroph. So autotroph makes its own food and the process is called photosynthesis, and this equation is very important, as you know. So 6CO2 plus 12H2O in the presence of light and chlorophyll will give rise to glucose, C6H12O6 and water and oxygen. So here I have written it as lungs. That is because of release of oxygen only plants actually, okay, they are called the lungs on earth, the green lungs on earth, and because they are producing food, so they are called the producers on the earth. And also I have written letter E here. That means these are the external factors. So what are the external factors for photosynthesis? Carbon dioxide, water, as well as sunlight. So if the definition is asked also about photosynthesis, you can write that, okay, manufacturing the glucose in the presence of simple inorganic compounds like carbon dioxide, water, light and chlorophyll. That becomes your definition also. Coming to the tests. So we have different tests here, okay, in this lesson where you have to learn. So you have the starch test, which is very important for all other tests. So starch test, what you do? So for all the tests, the central, okay, important thing that is needed is leaf. For all the experiments, the basic, okay, thing what you need is leaf. So leaf I have written in the center. So starch test, okay, you take a leaf, then alcohol or acetone you will put it in the water bath. Then leaf is taken out and then you'll add iodine, which will give rise to blue-black color. Okay, so that is the test for the starch, if you remember. Next, coming to the test for carbon dioxide, which is also called the Moll's half-leaf test. So here the leaf, half leaf is inserted into bottle with KOH. As I have written here, you can see it is KOH. Okay, so and then it is kept in sunlight. So after the period of exposure for sunlight for photosynthesis, you will take the leaf and you will do the starch test. So the leaf is negative. Which leaf will be negative? The part of the leaf which is in the bottle, because we know that potassium hydroxide, it absorbs of carbon dioxide that is present in the bottle. When there is no carbon dioxide that is present within the bottle, there cannot be photosynthesis. Hence the starch test will be negative for the part of the leaf that is present within the bottle. Okay, so that is what is written here. Next, for chlorophyll, you take a variegated leaf. Which leaf? Variegated leaf is leaf which will have, okay, white and also the green spots. And then you perform the starch test. You keep it in sunlight, okay, and then you will perform the starch test, and then, okay, the green portions only will develop the blue-black color, because green pigment, that is chlorophyll, is very much essential for photosynthesis. So these are the tests what you can, okay, just revise like this. Okay, and before all this, you know that one important point is any test in photosynthesis when you are doing, you have to keep the potted plant in the dark for 1 week to ensure that it is free from previously stored starch, which is very much important, okay, for your bits and all. That question can be asked. Then next, talking about, okay, the next structure what you have is chloroplast. So chloroplast, we know it has got three membranes, an outer membrane, inner membrane. Third membrane is put in the form of stacks, which are called the thylakoids. So three membranes. Grana region is where the thylakoids are there, the red color, okay, structures whatever I have drawn, those are the thylakoids that represents the grana region. In grana, you know that light reaction of the photosynthesis will take place, and there will be what are called light harvesting complexes will be present on the thylakoids, which are made up of your pigment molecules. If you remember chlorophyll A, chlorophyll B, carotenes and xanthophylls together, nearly 250 molecules will be making up your light harvesting complex, which will be playing a very important role in photosynthesis. Then next, talking about the stroma region. So the dots, whatever I have represented, the space that is present within the chloroplast is the stroma region, which consists of very essential enzymes that are needed for photosynthesis. So it will take part in the dark reaction. So in photosynthesis, you have light reaction and dark reaction. Dark reaction is independent of light. So that dark reaction will take place within the stroma with the help of the enzymes that are present. Dark reaction is also called the biochemical reaction. By the way, light reaction is also called the photochemical reaction, if you remember. Talking about the events in photosynthesis, so this is a brief diagram what I have drawn about the TS of the chloroplast. The two membranes, okay, and the red color structures are the thylakoids, which represent the grana region, and the dots region is the stroma, and the lines indicate the fretal lamellae and the membranes, inner membrane and outer membrane. Then talking about the events in photosynthesis, okay, we have principally three events if you remember. So I have just represented very easily like this: light plus chlorophyll, that is light is absorbed by the chlorophyll molecule and it will get activated, wherein I have represented a star on the top of the chlorophyll molecule. Then second step is conversion of light energy to chemical energy, Le to Ce I have written. Then next carbon dioxide is reduced, that is hydrogen will get added and it will get converted into carbohydrate, which is the third step, okay, conversion of carbon dioxide to carbohydrate. So these are the things what you have to remember about the autotrophs, which is very, very important and very, very easy also to remember like this. Talking about the other part of your lesson, that is heterotrophs. Heterotrophs, they depend upon other organisms. So we have three types: parasites, saprophytes and holozoic organisms. Parasites, they depend upon the host, as you have seen. I have written some examples: lice, leech, tapeworm and cuscuta, you have to remember, which is a plant variety, okay, that is a parasite. The next, talking about saprophytes, will feed on the dead decayed matter. So here we have written fungi, yeast and mushrooms as some of the examples. Then we have holozoic organisms. So we all humans are holozoic, wherein we ingest the whole food and digestion will take place inside the body of the organism. Those are the holozoic organisms. So much information we have to learn about holozoic organisms in this lesson. And most important example for this is amoeba. Even a short question for 2 marks can be asked. So amoeba actually is unicellular, it is eukaryotic, it is holozoic, okay. Intake of food takes place by pseudopodia and the process is called endocytosis. Talking about humans, in humans, okay, if you remember the different stages in digestion, first is ingestion, you take the food inside the mouth, followed by digestion, followed by absorption, followed by assimilation, and finally it will be the egestion. So these are the 5 stages in order. Sometimes they can be asked as a question for the MCQs. So do remember the order. Then next, talking about the alimentary canal, the different parts and what are their functions and the enzyme secretions. So talking about mouth, we have teeth. Okay, the dental formula is important. Incisors, canines, okay, premolars and molars is the order. Incisors, canines, premolars and molars. And the dentition formula I have written 2123 by 2123. Okay, and in the mouth, if you talk about, we have salivary glands, 3 pairs if you remember. So those are important, the parotid, submaxillary and sublingual. Okay, and they produce saliva, in which the enzyme is called salivary amylase, or it is called thiolin, which will be acting on starch and it will convert it into maltose. Talking about the second structure, esophagus, which is a long tube, it will just help in peristalsis. There is no enzyme that is present within the esophagus. Talking about stomach, stomach has got the gastric glands, which will produce HCl, mucus and pepsin and also lipase. HCl, basically it will kill the harmful bacteria that is present in your food. Okay, and also it will create an acidic environment for the action of different gastric enzymes. This is a very, very important question from this lesson. What is the role of HCl? Okay, talking about mucus, mucus actually it is lubricating, okay, agent, like we can say, within the stomach, and also it gives protection to the lining of the stomach so that HCl that is produced will not act on the walls of the stomach and digest it off. Okay, then next, talking about pepsin, it will act on proteins, okay, that your food contains, and it will break down into the peptides. Okay, then we have is lipase, which will act on fats and converts them into fatty acids. Okay, then next you have to remember about this structure, that is the pyloric sphincter. So pyloric sphincter will regulate the amount of food, okay, that is entering from the stomach into the duodenum. Okay, so this you remember. Next, talking about the next structure, that is small intestine, which is very, very long, some 6 to 8 feet length it will be. So small intestine, into that 2 secretions are released, that is, one is by the liver and other one is by pancreas. So the secretion that is given out by liver is the bile juice. Bile juice doesn't have any enzymes. This is also a very important question. Which juice will not have any enzymes? Bile juice. And its function is to emulsify fats. It will break down the fat globules into small globules, okay, for easy digestion. Okay, and you have to remember pancreas, it produces pancreatic juice, which consists of 3 enzymes, namely trypsin, amylase, and lipase. Trypsin acts on proteins, amylase acts on carbohydrates, and lipase will act on fats, converting them into the respective smaller molecules, that is proteins to amino acids. Okay, carbohydrates finally will be converted into glucose, and you have is the fats getting converted into fatty acids and glycerol. Okay, large intestine helps in absorption of water from the undigested food. That is the main function of the large intestine. Talking about rectum, it is the temporary storage for the fecal matter. Okay, from where finally it will be sent to the elimination center, that is the anus. So anus is controlled by the sphincters, if you remember, and finally the egestion of the undigested matter, the fecal matter, will take place through the anus. So this is how entire topic you can just remember through this flow chart. So like this, for every topic, if you start making the flow chart, then it will be very easy for you to remember the topic in a very easy way. Okay, so hope you maintain a separate book and start revising your topics like this. If you like my video, and if you want me to make okay, such type of videos for any other topics, do post in the comments. Okay, and click a like button and share it to as many as possible, and let them also subscribe. Thank you.